All right, I thought it'd be good to have a quick review of the genetic traits that you're going to see on the test, just give you a nice little quick background. This would probably be a very good presentation to have up and running while you're taking a test so you can refer back to it. So just a few terms, just so you remember what they are. Genetics is the study of heredity. What is heredity? The passing of traits from parents to offspring. And what are we passing? We're passing our genes, which are DNA that actually codes for a protein, which would be the trait. And we have different alleles of those genes. Sometimes you have two of the same alleles, or sometimes you have two different ones. Anyway, that would be like having freckles or not having freckles. Those would be two different alleles for the freckly face thing. Some other important terms to know. Dominant means you always see the trait if you have one copy of it. Recessive, you need two copies to see the trait. Heterozygous, you got one of each trait. One of each allele, I guess, to form your trait. Homozygous means that you have two of the same alleles for the gene. So you're purebred. You're either going to have two dominant or two recessive alleles. The genotype is the actual letters for the alleles. That's what you're putting in the Punnett squares. And phenotype is basically how those genotypes are interpreted. In other words, what do you see? All right, so in the test, you're going to have several of these different types of inheritance. So we'll just go over how do you know if it's this inheritance or that inheritance. So first one we'll talk about is autosomal dominant. So autosomal means it's not on a sex chromosome. It can be on any other chromosome. And if you have this allele, you're going to see that trait. No question about it. Generally, you only have two phenotypes. So that's a good way to know if you have it. You either have this or that. And the person just has one of those alleles, then they're going to see that dominant trait. So for these things, we generally use the same letters. The dominant for the uppercase and lowercase going to be recessive. You can make sure it's the same letter though. So if you're going to say hair loss and you decide to use H for hair loss, you would have big H if it was dominant and little h recessive kind of thing. All right. Autosomal recessive, again, we're not talking sex chromosomes. We're talking about the other chromosomes. So how do we know? Again, there's only two phenotypes. And for someone to see this trait, you need to have two of those alleles. In other words, no dominant allele. And the letters are basically the same as on the slide before. Incomplete dominance. In incomplete dominance, there'll be three phenotypes. So you'll have, if you got two of one gene, you will have this. If you have two of another gene, you'll have that. If you have one of each, you got something in between. There's a mixing, a blending in the heterozygous form. So the example we have is medium is between tall and short. So something in the middle. You can use two different letters. You don't necessarily have to. Just know that there is going to be a dominant one. It's going to be extreme size, and the other one's going to be the other side of things. If you got two of each of the alleles, then it is a blending. All right, codominance. Codominance means you see both alleles. So if you got this one, and that one, you're going to see both of them. So again, there'll be three possible phenotypes. It's the heterozygous that's going to show us. And again, we're using generally two uppercase letters because they're both dominant letters. So if we look at our example here of the flowers, if it was just straight dominant and recessive and you had a red gene, you're going to have a red flower. You're going to have two recessive white genes, so we'd have little r for white in this case. It would be white. Incomplete dominance would be you have one red, one white, so you get this kind of pinky color. And if it was codominant, as in this example, if you have a red and white gene together, you see both. So that's the codominant example, seeing both. All right, our last trait to talk about is the sex-linked recessive traits. We only talk about the recessive ones in this class. So sex-linked recessive means you need 2 copies. They will be on the X chromosome. So seeing something that says X-linked is probably a better term, because Y-linked doesn't really make sense. It only happens in males, so it's not a big issue. And really, Y-linked genes are all related to being male, so it has no compatibility with females. So females need 2 copies of these genes to be effective. Males only have 1 X chromosome, so they only need 1 gene. So this one is always seen in males more often. Hardly ever be seen in females. So again, when you look at it, there'll be a lot more males who have it than females. Sons can only inherit it from the mother. They don't get it from the father. From the father, they get the Y chromosome, so they can only get this one from mom. Some daughters do have it, like color blindness and stuff. They do have it. So basically, they got the father's X, and the mother could give her X, the bad X, and then she'd be colorblind. But generally, if the father has the trait, the daughter will be a carrier, because he only give her his X, his 1 X he has. So here we're going to use X and Y's. Make sure females are XX, males are XY. Don't screw that up. You got to have 1 of each to have a baby to fill in your Punnett square. So these are the possible genotypes. So in this case, we're looking at these little a letters on there. Sometimes people will just put the little a and leave the other one blank. They won't actually have a big A, but that just depends on the situation. All right, so that's the basic rules for the types of inheritance you'll see on the test. I recommend listening to my hints thing on the test, that video that I made. I think that'll help you out as well. So check out that one, and then you should be ready to go take that test. And again, if you have any questions, let me know. Make sure you do write down your Punnett squares. Don't try to do this without looking at a Punnett square. You will not do well. All right, that's all I got. Thank you. Goodbye.